fluorescence. For DNA barcode, the library is very important for species identification. The advantage of regional DNA barcode library is by reducing species diversity so that to increase the identification accuracy. Generally, the sequence was submitted to GeneBank and BOLD, but for GeneBank data, there are some limitations, such as it didn't demand to provide the vulture specimens photos. So this will cause the risk of misidentification. And also, it causes the difficulty that the vulture specimens cannot be used for morphological comparison. In most phylogenetic study papers, they only provide vulture collection information, not vulture photos. However, for both, there is very strict control and census. The vulture specimens information from collection, identification, and photos must be included to submit them. At the international levels, for the BOLD, most DNA library was established in Canada, United Kingdom, Australia, Norway, and Japan. And also, and some states in the United States, Mexico, and some other countries. This is the map of the barcode resource. From this map, we can get the following information. That is, only 71,000 plant species have them. In China, how about the DNA barcode library construction? I think the most important was on the medicinal plant. It was established by Professor Chen Shilin's team. This is the link. And also there is invasive plants and wood-treated wood species. And also by Dr. Li from Kunming Institute of Botany, they established Chinese plant DNA barcode identification platform. In this platform, there is more than 120,000 DNA barcode and more than 10,000 Chinese plant species. This is the link. In recent years, supported by Ministry of Science and Technology of our government, Chinese Academy of Sciences, there are many projects on DNA barcode. They focus on different regions, different plant communities. This is the link. From the board, we can find the DNA barcode resource. From this map, you can find that most of the DNA barcode is from the eastern region of China. That means the humid region. However, for plant, only 16% from plants is much less than the insect animals. This is a map on the global desert distribution. According to the definition, that desert means the annual rainfall is less than 250 millimeters. At global scales, the desert takes around 1/3 of the land area. So the biggest desert is in North Africa, Sahara Desert, and Central Asia. In China, the desert takes around 1/4 of Chinese territory. That means there's 2.5 million kilometers. It's a huge area. So in the Inner Mongolia and Xinjiang, Gansu, and some Qinghai province is a huge area. A desert plant, during their long-term adaptation, they have developed many convergent characters, such as fairies, smaller or reduced leaves, the flesh leaves, the spiny, etc. That makes them difficult to be identified through some morphological characters. And also, they grow in early spring to avoid the drought in summer, such as tulipa, papaya. For floristics of desert flora in China, through our census, there are about 1,400 species from 478 genera. From this map, we can find the biggest family is Asteraceae, Fabaceae, and Amarantaceae. And in this flora, the mono and oligo genera dominate. That means 90% of the genera are small genera. However, 1/3 the species are from the big genera. The big genera are Astragalus, Artemisia, Calagana, Salsola, etc. Compared to the east of China, the DNA barcode study on desert flora is very few. Now, I will introduce our study material and method. The first is flora species list compilation through studies the species list of Chinese arid region plant and many papers. After the name corrections through plant species data center. And last, we collected 1,816 plant species in this study. It is from 72 families, 386 genera, and include 890 species. It represents more than 80% genera and around 62% species in Chinese arid region flora. This is a protocol. We followed the protocol very strictly. That means the sequence was carried out from both sides by direction sequence. And we sequenced 4 barcodes, including 2 core barcodes, RBCL and K, and then 2 additional barcodes, ATS and ATS2. All of these informations, including vulture, specimens informations, and the sequence, was submitted to both systems. This showed the species, specimens number, sequence number. This data has been opened to public. Everybody can check it and download the sequence from both. In the data analysis, we used the 4 methods, that is, barcoding gap method, all-to-all blast method, tree-based method by establishing ML trees, and the similarity-based method by using the software taxonomy. By using the 3 barcode data combinations, we establish a community phylogenetic trees for all the desert plants. This is the phylogenetic tree. For 7 big genera, we tried to use the genome skimming method to get the chloroplast genome. and the data, complete plasmid genome, was submitted to GeneBank because in the old system, it didn't receive Fastone at this stage. This is the result. First, I will introduce the result for single locus. All the four methods showed similar result. That is, at the species level, RPCL showed the lowest species resolution. It is 51%. And the ATS demonstrates the highest species resolution is 72%. And the genus levels, RPCL is 76%. ATS is 80%. For the barcode combinations, the resolution increased. Among the 7 different combinations, RMI combinations is the highest. Its species resolution is 75.5%. And the genus levels, its resolution get 92%. However, among the 363 general studies, 18 general are not monophyletic. And these general are from Asteraceae, Brassicaceae, Fabaceae, and Amaranth. In the practice of species identification through DNA barcode, like monophyly is the main reason for identification failure at the genus levels. We found that the type Salsolia from Amaranthaceae is a big question. Many genera is not monophyletic. For example, several Salsola species clustered together was Haloxylum, Halogetum, Anabasis legion. However, and some Salsola species from the class with Heterosemonia. Another example is Artecus from the cluster with Astrotemenus fraticosus. That means Aster genus is not monophyletic. For the 7 big genera, for example, this shows the Calagana, this shows the Astragalus. Firstly, we showed that coding region dataset reveals the same tree with the complete plastomes. And another 5 genera I didn't demonstrate here. The topology is the same between the plastomes and the core barcode combinations, RMI combinations. The difference is only the hair support get from plastomes and CSRs. That's a little different from our expected result. This is the tables. From these tables, we can find that for 5 genera, Lactuca, Lactula, Lepidum, Silene, and Sigophyllum, they showed 3 different data sets. They showed the same result. The different result concentrate in Astragalus and Calagana. I guess the different reasons is firstly our sampling. The result calculated was only based on the species with 2 conspecific individuals. And then the closely related species were not sampled or calculated completely. So through this method, the resolution was inflated. Now I'll introduce the result for astragalus. Astragalus is one of the biggest genera in the world. There are more than 3,000 species. In China, we have 400 species. It's not only the largest genus in the desert flora of China. So it's very difficult to identify. For this mega genus, the identification to section level is very important in practice. However, due to in the specimens makings, they lost some characters such as the flower color, not floral color. Sorry, here it's flower color. Fortunately, in this study, there is a huge database that provided by Su and all. They have a good very study and provided 83 species. These species are from northwestern China. And then we download 12 species. In this study, we have 17 species and 26 individuals. The result shows that 9 species were identified successfully, including 5 species with 1 individuals. However, the species resolution is only 50. So the possible reason we guess is first, the species identification. Our vulture species might be misidentified, or, for example, this Ellipsoides and Alpinus. Another reason is that in the reference database, there is no reference for at least a section of series level, such as the Zhouquanensis, the 3 different sections. So we suggest for mega genus, the silk DNA barcode should prioritize a different regs, such as sections and series. For Kalagana, Yuan and all shows that Kalagana is not monophyletic, at least Halimodendron and Caulovaca were embedded in this section. And in our study, 3 species are from the same series. They cannot be differentiated by DNA barcode combination. But 1 species, Luana, was identified successfully. This means that plantstones improve the species identification for this species complex. However, it cannot succeed for another 2 species. So we suggest that in future, the nuclear genome marker is needed. That means the 3rd generation DNA barcode. And the last 2 minutes, I will introduce our work in recent 2 years about the desert in South America. So this map shows the Pacific coast in Peru. It is desert. And along the desert, there is 1 special early spring vegetation, the long mass. In the early spring, because there is fog from the sea, so it makes some special locality that face the sea could get the moisture from the fog. And there is a natural vegetation, the lomas. In the summer, this is a desert. And in the spring, it becomes a desert. And for the lomas in Peru, there are around 900 species. There are a few bigger genera, such as Bonana, Pallawa, and oxalates. In 2017 and 2018, we have 2 years systematic collection with the help from St. Mark's Natural History Museum. We collected 1,200 specimens and around 600 species. And we finished DNA barcode for 3 barcodes, Apical, Medicaid, and ATS2. We have uploaded them to both. This shows the result. That's all my result. During this study, we get a lot of help from many Chinese fluent and proven fluent. Thanks, all of them. Thank you.
Thank you very much to Xu Jun. We will share now the small presentation that I presented at another meeting, but I think you guys will also like it. So I'm going to share the screen now. And please let me know whether you can hear the talk.
Yes.
Okay.
Alina, we can hear it.
You cannot.
No.
You no. Len. Okay. Let me. Then I will transmit my from, but I am doing it from my computer. Let me see if there is a situation with Zoom. Let me see if Lisette is here. Yeah, nobody from from Cedia is here. You cannot hear my the audio. Okay, what can I do? Then I think I just talk if it's okay with you. Yeah. Let me go, and I just I'm not sharing it, right? No. PPP. Okay. Service talks. Finish. Yeah. I would just share the screen. And then, can you see now?
Yeah.
Yeah. Okay. So yeah. I'm going to just talk about the experiences we had and the potential that small institutions, not small institutions in countries where there is no graduate program, how the potential that we have to do promissory research with a contribution of undergraduate students. So we'll just go to the next one. So first, I'd like to remind you about the location of Ecuador. We are in the equatorial line, and we are a very small country with 283,000 square kilometers. Let me see if it's not. Yeah, okay. Ecuador is one of the countries that have two important hotspots: the Chocó Darien region hotspot in western Ecuador, and also the Andean, tropical Andean hotspot. We have a high diversity, so we have, in terms of percentage, per land area, we are a small country, but we have a high percentage of taxa from all organisms. And then in numbers of endemic species, for example, in amphibians, we have almost 300 species of amphibians. And so on, we have other numbers of species that are endemic to the country. Here are some examples of organisms that have been described just recently. So yeah, this one from Brito, it was small, the Andean mouse was discovered just in 2021. And then another bird that was discovered a few years. So we keep discovering species. As you might remember yesterday, our colleague from the coast, Javier, he and the same Katia, indicated about species they have described. So in terms of diversity of plants, we have a very, I think it's the highest number of species per 1,000 square kilometers. So we have about 65 species per square kilometer. And then we have more than 5,000 endemic species. Comparing to the other countries in the Americas, Ecuador is in the occupies the 5th place after Brazil, Colombia, Mexico. And as you know, these countries are much, much larger. And then we have more species than North America, for example. Unfortunately, we have 0 graduate programs of botany, even though we have a high diversity. We have some programs that have studies on biodiversity and conservation. From the 59 institutions, we have only 8 that talk about biodiversity. So my experience is from the time I was a professor at Universidad Técnica de Cotopaxi, which is located in the central Andean area of Ecuador. And I worked with students from the different faculties. So we work with the natural resources faculty and also with the administrative faculty. So the projects that we worked were in agroindustry, graphic design, community-based tourism, medicinal plants, and ecotourism. The localities where the students work were in the Cotopaxi province and in the Imbabura province in northern Ecuador. So the family that was the most common in the group from the various studies was the family Asteraceae. So we have 2 examples here, and we saw in China also in the dry areas or in the dry, it was the most common family. We know that it's the most diverse family in the angiosperms. The second group that have the highest percentage of taxa that were studied by students were the Fabaceae, and then we have the Lamiaceae, Amarantaceae, and so we have here examples of Fabaceae, like Inga edulis. It's called cotton sugar fruit, and then we have Lupinus, which is the source for chocho, which is like a lupin seeds that is eaten. So the first project was done with the student Carla Fuentes. It was a very interesting combination of graphic design and botany. So the purpose of her study was to describe botanically plants that were growing in an abandoned lot from her uncle. And she wanted, through the design of these species, to educate the local community about the importance of the study of these species, and also the importance and the need of protecting the native species. The next study was done by Andrea Pachacama, who did a study in markets in Latacunga. So she collected information about the plants, and then interesting in the markets, 60% of the species that she studied were exotic. A product of her research was this guide, and she included as co-authors to the members of the market who told her the information. And then she used GB for tropicals and then information from the herbarium to have her guide. The next study was done by Lisette Gavilanes. She worked instead in a indigenous community in the Cotopaxi province, and the purpose of her research was to provide the community with a guide that the community could use if there were tourists. This the mountain shown here, it's known as a touristic place. So what she did was to ask the community about uses. She did observations, and based on that, she provided information for the communities to share with others. The next study was done by Evelyn Zurita. She did the natural history study. What she did was in campus. The university has two campuses. One is called Salache. So she did the inventory of the families present in this place where there is potential activities for tourism, more like towards ecotourism. So she did observations about natural history, relationships between plants and animals and insects, etc. Her research found that 15% of the taxa were endemic, and she didn't find any exotics. And then another study done with students from the agro industry, they did a survey of presence or absence of mucilage in the various species that they went to the field in the Pichincha and Cotopaxi and Imbabura provinces. So they found about the same similar percentages of endemic and native species and very low percentage of exotic. So their purpose of this research was for them to provide a means of identification of the taxa with potential use in the industry for mucilage. So what they found is here we have several species. It was recorded for the first time in Ecuador of these species. Actually, they had a lot of trouble finding literature about mucilaginous species. So as a summary of these studies, we found that 57% of the species that they studied were natives, excluding the endemics, and 14% were endemic species, and 28, almost 29% were endemic. Were exotics. So depending on the study, the percentage of each one of these three categories would change. Now, the problem is that now we are in the Anthropocene, so we are really in a bad, we have a bad future. Yeah, if we see that temperature is increasing very badly, the information that they are getting is to find potential applications of plants and potential means of protecting these species, because as you know, we are at a very high risk of losing taxa, especially in the tropics. So for the future, as I mentioned, the conservation, education, research in Ecuador right now depends mainly on undergraduate students. So that's all. I'd like to thank everybody, and if we have questions, we'll be here to answer them. Xu Jun, and myself. Thank you. So if we don't have any questions, inquiries, or anything, in that case, we will continue with the program. As right now, it's 7:47. Let me see, according to the program, we will have the participation from Xu Jun, from Limin Lu. So I will share the biography of Limin, and so Xu Jun could introduce her to the speakers. Let me see. Okay. Sure. Thursday to Limin Lu. Yeah. Okay. So I will share the screen now. So Xu Jun. If you please could introduce Limin Lu.
Okay, it's my great pleasure to introduce the young scientist in China, very distinguished young scientist, Dr. Limin Lu. Dr. Limin Lu works at the Institute of Botany, Chinese Academy of Sciences in Beijing. Her research interests focus on plant tree of life reconstruction and biodiversity conservation. She has made several original contributions in evolutionary history of angiosperm flora China and the phylogeny and the biogeography of grape family, Vitaceae. She has published more than 30 papers in journals such as Nature, National Science Review, and Cladistics as the first or corresponding author. And the research in Nature has been selected as one of the 2018 top 10 scientific advances in life sciences of China. She has been successfully funded by the Young Elite Scientist Sponsorship Program by China Association for Science and Technology and the Young Innovation Promotion Association Chinese Academic of Sciences, and the Excellent Young Scientist funded by National Natural Science Foundation of China. In this conference, the topic is on the temporal and spatial comparison of angiosperm diversity between East Asia and North America. It's my great pleasure. Please, Limin, it's turn to you.
Thank you, Professor Ge. So I will share my screen at this side. Can you hear the voice?
Yeah.
Yes.
I'm from the Institute of Botany, Chinese Academy of Sciences. First of all, I would like to thank Professor Xue Junge and Alina for providing me such a good chance to communicate with botanists from both China and Ecuador. It's also my pleasure to share our research progress here. The title of my talk today is the Temporal and Spatial Comparisons of Angiosperm Diversity between Eastern Asia and North America. As the lovely poem sings, all things bright and beautiful, all creatures great and small, life forms in all kinds of magical shapes and varies from place to place in a highly non-random and predictable manner, making our Earth a colorful world. It's our honor to be a scientist to study the variation of life on the planet. In our research of life science, we are devoted to explore the following fundamental questions: the origin of life, their relationships, and the distribution patterns of biodiversity. As early as 200 years ago, scientists had discovered the distribution of biodiversity on Earth is far from even. The tropics contain many more species of both plants and animals than an equivalent area of the higher latitudes. This is the well-known latitudinal diversity gradient phenomenon, which can be simply described as biodiversity increasing from the poles to the equator. My interest is to test if the phenomenon of a latitudinal diversity gradient is a general pattern across the world, and
to investigate the underlying mechanism of such uneven distribution. Besides the uneven distribution across geographical regions, the speciation and extinction rates of species also vary through geological periods. For instance, previous studies have suggested that about 80% of ordinal and interordinal angiosperm clades have diversified during the early to middle Cretaceous, as the pink shaded regions shown in the slide. As for conifers, the lineage evolution was punctuated by 4 known mass extinction events over time. It's thus even more interesting to investigate the uneven distribution of diversity from both the temporal and spatial scales. An important question is how can we get the temporal and spatial distribution of biodiversity? Tree of life has been considered a bridge connecting biological disciplines, such as molecular biology, ecology, genomics, and bioinformatics. Then, what is a tree of life? The term tree of life was first proposed by Charles Darwin in 1859 as a metaphor for describing phylogenetic relationships among organisms. A dated tree of life can provide divergence times for all taxa and ancestral nodes involved, which can help us to document the diversity patterns from a temporal scale. Then how can we incorporate tree of life into biodiversity research? Here, I will introduce another concept, phylogenetic diversity, hereafter shortened to PD. It was proposed by Faith in 1992, defined as the sum of branch lengths connecting all taxa in a region. Since the branch length indicates evolutionary distance, recent studies have suggested phylogenetic diversity to be a better measure of biodiversity than only counting and comparing species numbers. Here is an example. We have 2 plots that both possess 3 species. In plot A, the 3 species are closely related, and their sum of branch length is low, while in plot B, the 3 species are distantly related, and their sum of branch length is high, and thus possesses higher phylogenetic diversity. Not considering other facets of diversity, protecting plot B can obviously conserve more ancient evolutionary history than protecting plot A. As PD is usually positively correlated with the richness, a standardized PD was proposed to exclude the accumulative effect of taxonomic richness. A significantly higher standardized PD usually suggests ancient evolutionary history, while a significantly lower standardized PD often represents recent diversification. China, home to 30,000 angiosperm species that cover nearly 10% of angiosperm species worldwide, is ideal for investigating the spatial and temporal patterns of plant diversity. Whether areas within China serve as cradles or museums remain unclear, as floristic components of Asian origin and of recent diversification have both been discovered. In the slide, photos on the left present some taxa of Asian origin from Eastern China, such as Megalitia, Euptalia, Chloranthus, and Lithocarpus. Photos on the right present some taxa of recent diversification from Western China, such as Rhododendron, Pedicularis, Gentiana, and Sausuria. We investigated the temporal and spatial diversity patterns of the Chinese angiosperm flora using a dated phylogeny of 92% of genera and detailed spatial distribution data. Our dated phylogeny included more than 5,000 species, representing 2,665 genera native to China. We validated the available fossils and selected 138 collaborations for dating analysis. We assembled a distribution database that contained 1.4 million occurrence records. The distribution data were at county level. We then divided the map of China into 100 kilometers by 100 kilometers grid cells to minimize the sampling bias of unequal sampling areas. Our divergence time estimation shows that about 66% of the angiosperm genera in China didn't originate until the early Miocene. From the table, we can see about 48% of angiosperm genera diverged during the Miocene, suggesting that Miocene is an important and key epoch for the diversification of angiosperm flora in China. Mapping the mean divergence times of all genera revealed a transition belt that coincides with the modern 500 millimeter isoline of annual precipitation, which marks the boundary between humid to semi-humid and arid to semi-arid areas. The angiosperm flora of Eastern China bears a signature of older divergence and higher phylogenetic diversity. By contrast, the angiosperm of Western China shows more recent divergence and lower phylogenetic diversity. Moreover, genera occurring only in Eastern China are distributed throughout the tree of life, with both anciently originated clades and recently diverging lineages. However, genera that occur only in Western China are predominantly members of only a few clades, such as Apialis, Australis, and Brassicalis, most of which have much younger divergence times than the major clades of Eastern China. These lines of evidence support a younger West and an older East of the Chinese angiosperm flora. Eastern China and Western China also show distinct divergence patterns for herbaceous and woody plants. In Eastern China, of the 1,000 genera that occur only in Eastern China, 39% originated before 20 million years ago, and 61% arose more recently than this. Both herbaceous and woody genera diverged at similar rates throughout geological time. In Western China, however, of the 100 genera that occur only in Western China. 76% originated in the past 20 million years ago, and only 24% originated before this time. In Western China, a much higher percentage of herbaceous than woody genera has originated since 30 million years ago. From the mapping of the mean divergence times of herbaceous and woody genera, we can also indicate that Eastern China represents a floristic museum, and Western China an evolutionary cradle for herbaceous genera. For the woody genera, however, Eastern China has served as both a museum and a cradle. From the above results, we find that the spatial divergence and diversity patterns of angiosperms detected here do not precisely reflect the latitudinal gradient in China, with the mean divergence time and phylogenetic diversity decreasing from southeast to northwest. Our regression analysis showed the mean annual precipitation and mean annual temperature have a higher explanatory power for the mean divergence time patterns. Our results indicate the importance of water and temperature in limiting the dispersal of species from humid and warm regions to drier and colder areas. The effects of topography with a pronounced altitudinal gradient increasing from east to west, and the monsoon climate in Eastern Asia are so extensive that the decreasing temperature and precipitation gradients from southeastern to northwestern China are not consistent with the latitudinal gradients, as might be expected in flatter regions. Moreover, our regression analysis show the mean annual precipitation and mean annual temperature have a higher explanatory power for the mean divergence time of herbaceous genera than of the woody genera. These patterns may reflect the heterogeneity in rates of evolution between herbaceous and woody lineages. Herbaceous plants are well known to have higher substitution rates owing to their shorter generation times, which perhaps allow them to respond more quickly to environmental changes through increased genetic divergence and speciation rates. We also calculated the top 5% highest phylogenetic diversity and standardized the phylogenetic diversity to locate hotspots of diversity from the phylogenetic perspective. We found that phylogenetic diversity hotspots are mainly located in several provinces of southern China, such as Guangdong, Guangxi, Guizhou, and Hainan at the genus level, and mainly Yunnan at the species level. These areas are also hotspots for threatened plants in China, as inferred by previous studies. Our results suggest the need to establish more connections between existing natural reserves and national parks that span provincial borders to conserve plant lineages of both ancient and recent origins. After completing the China project, we would like to investigate the evolutionary history of flora of the Northern Hemisphere. Fortunately, we were involved in a dimension project hosted by Professor Cheng Xinfu from Zhejiang University and Pam Sotis from University of Florida. The project focused on exploring how historical constraints, local adaptation, and species interaction shape biodiversity across an ancient floristic distinction. Funded by the project, we investigated temporal and spatial comparisons of angiosperm diversity between Eastern Asia and North America through working closely with the team members from both China and the USA. Eastern Asia and North America share similar geographical environments with comparable latitude, land area, and climate, except that North America has a coastal line in the west. The two regions were once parts of the same ancient landmass, Laurasia, and their extant floras were hypothesized to derive from the once widespread Botanical flora that spanned the northern hemisphere in the Paleocene. Even after the Paleocene separation of Eastern Asia and North America, floristic exchanges between the two regions have continued via land bridges, such as the famous Bering Land Bridge and North Atlantic Land Bridge. Despite floristic similarity between Eastern Asia and North America, the number of vascular plant species in Eastern Asia is at least 1.5 times larger than that in North America. So, a detailed comparative study of the floras in Eastern Asia and North America affords an ideal test case for exploring processes that contribute to this highly uneven distribution of biodiversity. However, much of the earlier work focused on comparing disjunct lineages or taxonomic diversity between the two regions. Therefore, a broader comparison of floristic diversity between Eastern Asia and North America is desirable and may shed new light on how the two floras were assembled. China and the USA cover almost half of the land areas of Eastern Asia and North America, respectively, and harbor much of the plant diversity in the two regions. Besides, abundant occurrence records are available in a consistent format within each country, and genetic data are available for many species in the two countries. We herein explore the floristic differences between Eastern Asia and North America using the well-studied angiosperm floras of China and the USA as exemplars. We newly generated a dated phylogeny of angiosperm floras from China and the USA, which includes more than 9,000 species, representing 3,700 genera, and covers more than 90% of native genera from the two countries. We estimated the divergence times using 139 calibration points in TRPL and assembled about 3 million distribution records to explore floristic differences between Eastern Asia and North America. We found that, in general, China possesses both higher richness and phylogenetic diversity for angiosperms than the USA, and most lineages, 46 of 58 orders investigated in the angiosperm tree of life, have contributed to the PD anomaly favoring China. The figure on the left is a summary phylogeny of angiosperms from China and the USA at the order level. The bars on the right represent generic richness and phylogenetic diversity of each major lineage in China and the USA. 10 dark circles on the terminals of the tree correspond to the top 10 orders with the highest PD differences between China and the USA. Photographs of representative species of these orders are shown on the right. According to our earlier study, we divided both China and the USA into East and West by the 500 millimeter ISO line of annual precipitation. As the dashed line in the maps, we calculated numbers of angiosperm genera that originated during each 5 million year period in China, the USA, and their eastern and western parts. Red solid line represents Eastern China, red dashed line represents Western China, blue solid line represents Eastern USA, and blue dashed line represents Western USA. From the figure on the left, we can see Eastern China has more genera that originated during each geological time span than the other three regions. However, the pattern of divergence times within the USA is more complex than within China. The East USA has more genera that originated during the Paleogene than the West, but the West then surpassed the East after the Middle Miocene. We also conducted analysis for woody and herbaceous species separately. Figure on the right shows that the herbaceous genera have similar divergence patterns as all genera, with the contrast between Eastern and Western USA even more obvious. However, the woody genera in the Eastern USA exhibit consistently higher divergence during each geological time span than those of the Western USA. This result demonstrates that herbaceous lineages from the Western USA have experienced rapid diversification since the Middle Miocene, perhaps in response to the recent orogeny and an increasingly arid climate in these areas. We calculated numbers and the proportions of genera that originated before the Miocene and after the Miocene to identify divergence signatures for floras in China and the USA. We found that Miocene is a key period for diversity of both China and the USA, each with more than 50% of diversity generated during the Miocene. China has preserved a higher proportion of genera that originated before the Miocene than the USA. We also mapped the distribution of genera originated before and after the Miocene to show where are the older and the younger lineages distributed. From the figure on the right, southern China is a center of diversity for both genera of pre-Miocene origin and recent origin. In the USA, pre-Miocene genera are mainly distributed in the southeastern coastal areas, while post-Miocene genera are mostly distributed in the southwest. This result suggests that southern China bears a signature of both old and new diversification, which is congruent with our previous study. For the USA, the Eastern USA has preserved more Asian lineages, while the Western USA holds more young lineages. In a spatial scale, both the floras in China and the USA share the signature of an older East and an younger West. And China shows greater east-west deviation in divergence times and diversity than the USA. The patterns within the USA are more complex than those of China, with the East possessing lower genus richness, but higher PD and standardized PD than the West, suggesting more Asian lineages in Eastern USA. Increasing number of studies indicate that conservation efforts should focus on the protection of regions with high phylogenetic diversity rather than species richness to better prepare for uncertain environmental changes in the future. The rough assessment of phylogenetic diversity for Chinese and USA floras allow us to make conservation recommendations across portions of two continents. When all genera in China and the USA are treated as a single sampling pool, southern China has areas with both the highest 5% PD and the highest 5% standardized PD. In agreement with our earlier study, the Eastern USA has the top 5 standardized PD, and the hotspots are all part of the North American coastal plain. The 36 biodiversity hotspots recognized by Conservation International. The species level analysis didn't detect any phylogenetic diversity hotspots in the USA. We can see from the figure, hotspots are all distributed in southern China. These results may not be surprising, given that generally represent larger units of genetic diversity and deeper cladogenesis in a tree of life. Therefore, different patterns of phylogenetic diversity at the species level indicates different histories and processes of the two floras in relatively recent times. We also treated the text of each region as an independent sampling pool. We find that at the genus level, phylogenetic diversity hotspots are more centrally located in southern China. In the USA, phylogenetic diversity hotspots are mainly in the east and the southwest. Phylogenetic diversity hotspots at the species level are largely consistent with those at the genus level, but areas with the highest 5% PD are more concentrated in the Hengduan Mountains of China and the southwestern USA. The protected land areas are highlighted in green on the maps. We can see protection zones in southern China and the eastern USA are fragmented, largely due to human activities, while western China and the western USA are covered by relatively larger areas with some measure of protection. Our study therefore suggests that more conservation areas in southern China and the eastern USA are needed to improve plant conservation efforts in the two countries. Moreover, the phylogenetic diversity analysis in this study supports the recognition of the 36 global biodiversity hotspots in the Northern American coastal plain. We therefore highlight that phylogenetic diversity is a key metric in biodiversity conservation. On the basis of the above results, incorporating geological and climatic evidence, we speculate that diversity anomaly between China and the USA might be largely driven by regional orogenic activities and climatic changes in the west of the two countries. Our team is keeping on exploring the mechanism of uneven distribution of diversity between the two regions. Hopefully, I can share the new results with you in the near future. I sincerely thank my supervisors, Professor Zhiduan Chen and Professor Jun Wen, for their guidance and patience throughout these years. I'm grateful to our collaborators, in particular, Pam Sotis, Doug Sotis, Stephen Smith, Russell Barrett, Lingfeng Mao, and Youhua Chen for their insights and inspiration, and to our team and excellent students for their creative work. Finally, an advertisement for our team. Our team is a newly established big team in the Institute of Botany, which is named Plant Big Data and Biodiversity Conservation Research Team. We welcome young talents having interests in plant big data, tree of life reconstruction, and biodiversity conservation to join us. Please feel free to connect me or our team leader, Professor Zhiduan Chen. I also list 2 major scholarships in China for international students. That's all for my talk today. Thank you for your attention. I'm ready to take any questions. Thank you.
Thank you, Li Min. There is one question in the chat room.
Okay, can you hear me?
Yes,
Can you hear me?
yes.
Yeah, because I needed to change the microphone for the video. Okay, so do you think collecting bios can interfere with results of morphological diversity in southern China? Okay, I think our results will not be biased by collection bias, because at a species level, so there may be more bias between Eastern and Western China. But our study is at the genus level. And we also covered more than 92% of the genera native to China. So we don't think our results will be biased by the collecting bias. Thank you.
So more question?
Yes, Li Min, great talk. I have some simple questions. So my first question is, during the comparison of China and USA, the species richness changed. Can you please go to that slide? That we can see there is a dramatic decrease for the PD for East China for the recent 5 million years ago. Why is it happen?
So maybe I should share my slides.
Yes, please.
Okay.
Oh, another very basic question. Maybe I missed the PD. I understand PD, but what's a different standardized PD meaning?
Okay, wait a minute. I will share my slides. Can you hear me?
Yes, but there is no.
Yeah,
No
there's
screen.
some problem of my. Okay. So wait a minute. Here it is. Okay, can you see the slide?
Yes.
So maybe I show you the standardized PD. So here. So actually it's the SES PD.
Oh, yeah. So how what's the, I mean, more common meaning, a simple meaning for this? It means because you classify different protection area based on both PD and PDSES.
Yes,
so PDSES means you can, I mean, from which perspective to understand the standard PD?
Okay, so still use the two plots here.
Yeah.
So for the example, both plots have 3 species, right? But so for the PD, if it can be affected by two factors, it can be really because of other evolutionary history like this. But it can, if the two plots have different number of species, like if plot A have only 3 species, but plot B, if plot B have only 3 species, but plot A like have 30 species. So if we calculate PD using the sum of branch lengths, still plot A can have a higher PD, right? So that means it can be affected by species number.
Oh, yeah. So it's standardized by the total species number.
Yeah, so it's the standardized PD will reduce the influence of species number. So,
but one of your conclusion is we prefer to use PD.
Actually, we use both. So I show you some. So like this one, we have a pattern of richness PD and standardized PD. So you can see the patterns, richness and PD are highly correlated, right? Even, especially in China. In USA, actually it's still highly correlated. But we can see from richness, actually Western USA has a higher richness. But for PD, actually, both Western and Eastern USA, it's at a similar level. But for standardized PD, after we reducing the effects of species number, we can see Eastern USA has
That's the slide that I asked you the question.
Yes. So this means it's like it conforms. It conforms. Eastern USA has possess like ancient evolutionary history. So So this is the most important conclusion in our study because we tested this from both temporal and spatial scale. Because we can also see this from this temporal.
That's my second question. Is the red line, the red solid line? From about 7 or 8 million years ago, it's a dramatic decrease. What's the reason? Maybe.
And so this one?
Yeah.
So I think this is at the genus level. I think more correlated with the global temperature, maybe I don't know. Maybe
the extinction.
What?
Some extinction.
I don't know. Because this is the genus level. So in our other studies, we can see the genus level. It indeed has a decline in recent periods, but at the species level, it's still increase.
Okay.
Yeah.
Oh, thank you.
Yeah, so I think maybe correlated with the extinction.
I don't know. I just feel curious.
Yeah.
Huimin, there are 2 more questions in the chat room.
2 more questions. Which one? I'm getting confused.
The last 2. One is from Sophia.
From Sophia.
And another from Mac.
Do you think that the molecular sampling, the global results? Okay, first I answer the question from Sophia. So you mean the molecular sampling could change the parameter PD and consequently the global results. I think the sampling type of genome denser, poor. So I think the molecular sampling is important, but you have to have a higher, coverage of the areas you studied. Like for our China project, we sampled more than 92% of genera. For the comparison between Eastern Asia and North America, we also sampled more than 90% of genera native to areas. So you need to have higher coverage of the sampling, and for the resolution of the tree, I think it's also it may also affect the results, but maybe not that much, because for now, if you use nuclear and chloroplast for some specific clades, you may got different positions, but for most of the genera, I think the relationships are congruent. Let me check for Alford. You're using general for many because what? I think greater morphology changes occurred. Okay, so another question from Mac Alford. Yes, indeed, genus actually genera is an how to say, arbitrary in terms of phylogeny. But I think that's what we can do for now, because we do not have a species level data. Our team has been doing a lot for generating a species level phylogeny for the Chinese vascular plants. For the first version, we sampled 92% of the genera. For the second version, we included more than 95% of genera and about 44% of species. So now we are still trying to collect more species, but it's hard. I think the third version, maybe if we can sampling more than 60% species, that will be great. So I think for the species level analysis, there's still a long way to go, especially at the global scale. So I think for a genus level analysis, that's what we can do for the data we have for now. Thank you.
Okay, thank you, Li Min. Very good study. Thank you very much. Let's
okay,
move
thank
to
you.
Next
Thank you.
speaker.
Okay. Thank you very much, Li Min. Very interesting. So please, if you could include your email so people could contact you. It's been a very interesting talk, and people would like to would continue to communicate with you via email. So we will continue with the next speaker. First, I'm going to share with you. So the next speaker, I will share the biography of our next guest. So it's my pleasure to introduce you to Hugo Valdebenito. Dr. Hugo Valdebenito earned his PhD in botany from the Ohio State University in Columbus, Ohio, United States in 1990. He subsequently served as head of botany at the Charles Darwin Research Station at the Galapagos Islands. And since 1994 has been a professor in the College of Biological and Environmental Sciences at the Universidad San Francisco de Quito in Quito, Ecuador. His interests include teaching and research in botany, ecology, and wildlife conservation on the flora of the Galapagos Islands and mainland Ecuador. He has authored more than 30 scientific papers and participated in several national and international meetings. Currently, he is the director of the Herbarium of Botanica Económica del Ecuador, QUSF. His talk would be about the flora of the Galapagos Islands. So welcome, Hugo, and then I will stop sharing the screen.
Okay, okay, thank you, Alina. I cannot open my camera, my video. He says that the host has stopped my camera, so I cannot. You cannot see me. But anyway, that's fine. I mean, don't worry. Yeah, you cannot initiate your video because the host has stopped
Hugo,
the function. Yeah.
Well, you should be able to do it now.
Okay,
let me check. Gave you access.
Yeah.
The right
now.
to access.
Yeah.
Wonderful. Thank you.
Yeah. Yeah. Hi. Hello. Okay. Thank you, Alina. It's a great pleasure to be with you and share some of the advances and generalities about the flora of the of the Galapagos Islands. I had the opportunity some time ago to live and work there. So I have some experience, and still I have some projects in carry on in the Galapagos Islands. So what I'm going to do now, I know I am going to to share my screen and start with my talk. Okay, can you see my slides? Yes, right?
Yes.
Yeah, okay.
Yes.
Okay, great. Okay, so what I'm going to do, as I said before, I'm going to talk a little bit about actually not just taxonomic topics, but also more like a generality about the Galapagos flora, from taxonomic point of view up to the conservation of the Galapagos flora. So in order to do that, first, I'm going to give a brief introduction about the environmental setting of the Galapagos, like where it's located, the climatic conditions that are going to determine different vegetation zones on the islands. Then I'm going to talk a little bit about the origin of the Galapagos flora, the geographical attachments or origins to the mainland or continent. And also, I'm going to talk a little bit about the endemic, native, and introduced species. Yes, unfortunately, we have people living there, and that means, in other words, introduction of not only plant, but also animal species. And then I'm going to give you some very interesting features about the Galapagos flora, specifically addressing the endemic genera, regarding this harmonic composition, adaptive radiation, and examples of island giantism. And we are going to end up talking a little bit about the conservation of the Galapagos flora regarding some efforts that some organizations are doing in Galapagos in order to preserve the endemism down there. So let's start with the environmental setting first. Okay, as you may all know, the Galapagos Islands is in front of the Ecuadorian mainland and 1,000 kilometers off the Ecuadorian mainland. And it consists basically of a set of islands plus an marine reserve. In other words, Galapagos is interesting not only because of the endemism that is occurring on the islands, but also in the ocean, in the territory along the islands. And here is the set of islands. There are more or less 12 major islands. And the oldest one, as we're going to see, are in the eastern part of the archipelago, and the youngest ones in the western part of the archipelago. The larger island here is Isabella, and the most, how can I say this, active island from the tourism point of view is Santa Cruz, is this one, which is located in the middle of the archipelago. Now, what is important here, or what I have to mention, is that the Galapagos National Park is what is in red and, sorry, in green here. That means, in other words, that 97% of the land area of the islands is protected under this figure of national park. And 3% is an inhabited area by more or less 35,000 residents. So in other words, there are 4 islands inhabited, yeah, but more or less in total, 35,000 residents. Now, the Galapagos Islands are oceanic in origin. That means, in other words, never have been attached to the Ecuadorian or to the mainland. And the youngest islands are on the western part of the archipelago. And there is an hotspot or a volcano producing area, which is producing, yeah, originating islands. So what we have is more or less this figure. We have the hotspot here producing volcanoes, in other words, islands. And since the Nazca plate is moving at 7 centimeters, the rate of 7 centimeters per year towards the mainland or towards America, you have that the youngest islands are in right there in the hotspot where they are originated. And of course, since this Nazca plate is moving towards the eastern part, the oldest ones are in this area. Yeah, they are further away from the hotspot. So in other words, what you have is this. You have the volcano, in other words, the island. This is moving up. This is moving towards the eastern part. And so this island, the oldest one, are going to finally ends up sinking under the ocean. And here it shows you the age of the islands, more or less, from the oldest one, 3 million years, and the youngest ones, more or less 700,000 years. And as you can guess, this is very important for phylogenetic studies of the origin of the different genera and species in Galapagos, because you can set, right, a really nice timelining from the oldest islands to the youngest ones. So in other words, you can see more or less how the flora, regarding, for example, just one genus, how has been evolving throughout the years in the archipelago. Now, regarding climatic conditions, these are very important because are going to determine different flora according to the elevation of the islands. So in Galapagos, we have mainly 2 climatic seasons or conditions. What we call a rainy season from January to June, and a dry season from July to December. The characteristics of these seasons is that the very first one is hot and humid, and the second one cool and dry. In other words, the vegetation somehow has to adapt also, not only to the altitude above the sea level, we're going to see a figure right now, but also to these climatic conditions during the year. And also in Galapagos, it's very interesting because we have several oceanic currents, like this one, El Nino, for example, is a warm current, but also we have cold ones. Like this one coming from Antarctica, the Humboldt current, or the Cranwell current. So the archipelago also have some changes throughout the year, or I mean during the year, according to these currents. I'm talking about more humidity or is more dry. And this is more the very first one on the top here. This is the condition of the vegetation zone that we have. I mean, we have more or less seven different vegetation zones, from what we call the littoral zones up to in the summit. It's more humid, the fern sedge zone. We're talking about the summit more or less 900 meters. Yeah, so you go from 0 to 900 meters. And since the humidity increases, also the vegetation is different. It's differentiated on these different ecological zones. However, this is very interesting. There are some islands where at least one, the largest one I showed you, the Isabella Island, the summit is more or less at 1,500 meters. Yeah, and what happened there is that the summit, yeah, or the top of the islands, it's above the inversion layer. So this area is quite dry, very similar to what you can see here in the dry zone. In other words, you are going to you are going from the lowlands, very dry, very humid, and suddenly, oops, here on the top, on the summit, it's dry again. So there are some species like cacti, for example, that are present in both areas. I mean, on the top, on the summit, as well as here in the dry zone. Now, this is very interesting. We are looking in one point. You stand in one point in the islands. Let's say, for example, here, yeah, you are there, standing up. And you take a picture towards the northern slope in this direction. What you are going to see is something like this. And then you turn around 180 degrees and you're going to see this. It's very dramatic change. It's more humid. In other words, the slopes facing the south are more humid than the slope facing the north. It's more or less something like this. The winds and the clouds are coming from the south. So this area, this slope, is more humid than the other side where you are going to get just this grainy shadow. Okay. Now, talking about the north and south side, of course, you can differentiate distribution of sometimes the same species. They have different distribution according to the altitude and according where if they are growing in the north on the south side. For example, let's take one quick example. The Opuntia echios is present in not only in the north side, but also in the south side. And as you can see here, for example, the Opuntia echios is distributed from 0 meters up to more or less 400 meters. Okay, this is altitude above the sea level of the islands. So that goes up to more or less 400 meters. However, in the slope that facing the south, the Opuntias just go up to probably 100 meters. Yeah, so the difference in vegetation between what you have in the north and in the south is quite striking. It's very, very different. So you end up with something like this. See, from the lowlands up to the highlands, it is very dry in the lowlands, and then in the highlands it's quite humid. Now, talking more specifically about the vegetation zones, what we're going to have is this. I mean, first, the vegetation zones are determined by 3 major environmental factors, like the age of the islands. Of course, the rate of erosion is important here, the orientation and the altitude. These are factors that are going to determine these different ecological zones or ecological areas that I was talking about, from the littoral arid transition zone. The transition zone, of course, the name says that transition, right? Well, you are going to see vegetation not only present here in the Scalesia zone, but also in the arid zone. Then the brown zone, Myconia, a fern sedge zone. Of course, the number of or the presence of these ecological zones is going to be different according to the altitude of the specific island. Some islands, of course, are lower. I mean, maybe they are going to up to 400 meters, no 900, but 400, 300, 200 meters. So not all these ecological zones are going to be present on every single island along the archipelago. So it's taking a quick look to the littoral zone. This is more or less the landscape in that zone, where the mangroves, succulent herbs species are quite abundant. And of course, these species are adapted to the dispersal by the oceanic currents, okay? And of course, there are few endemics because actually many species coming from the continent, yeah, are going to reach this place. Now, these are pictures of some of the species present in this littoral zone. And then we go to the arid zone. It's very beautiful, like what you see here, these succulent species, and they change in color according to the age. Yeah, the youngest, so the younger ones are more green, and as they get older, they start losing some pigments like chlorophylls. So you are going to see more like carotenes and other pigments. But anyway, it's very beautiful. This arid zone is characterized mainly by the presence of the opuntias or cacti in general. And what's very important here is that this arid zone has the highest number of endemic species present in Galapagos. Around 70% of the species, the endemic species are present here. And that is because the islands have been exposed to this dry condition for a longer period of time. Yeah, 10,000, 15, 20,000 years ago, we still had this arid zone on the whole the islands. But then have been changing according to currents, according to different geographic modification, and on and on. So we end up now with that the arid zone is just in the lower part of the island. Some of the species are here, Pisidia, Bursera, and of course the cacti are the most common plants like Opuntia echius, Brachycerus, and Jasminocerus. By the way, these two genera are endemic to the islands. Here you have some pictures of them. Then the transition zone, as we are going up, right? Transition zone, we are here more or less. So it's this ecological zone is between the Scalesia and the arid zone, as I mentioned before. As here, there are some of the representatives. Pictures are here: Tunifortia, Clerodendrum, Prosopis, among other, of course, other representative or species. Now, this zone, the Scalesia zone, is very interesting. Is where the Scalesia lives or is distributed here. The Scalesia is an endemic genus. I'm going to explain just a little bit about this. But it's an endemic genus with several species. Yeah, it looks like, how can I say this, a broccoli, perhaps, right? And is characterized, I said, by Scalesia, specifically by Scalesia pedunculata. The common name also is daisy tree, usually, and is present in Santa Cruz and Santiago, two of the islands, mainly. Other islands also, but just remnants of them. Yeah, unfortunately, this area or this ecological zone has been used for agricultural land since it's very suitable for agriculture. It's very humid. Some of the representatives of the Scalesias, and then we are here in the brown zone. It's very interesting, this zone, very characteristic. And this zone is characterized by epiphytes, and they turn brown during the arid, let's say, season, yeah? And some of the species that are present there are here, Santoxylum, Agnistus, and others. Here you can see some of the specimens, some of the species, And then we are in the Meconia zone. And the Meconia zone is characterized by this endemic species, by Meconia robinsoniana. Is endemic, of course, is endemic to the islands. And of course, it's a shrub, well, you know, the genus, right? And fortunately, has gone very strong alteration due to agriculture. And then we are in the summit, and the fern sage zone. It's characterized by ferns, by grasses, and also by this endemic tree fern, Cyathea weatherbiana. And it's this one, yeah, and it grows, as I said before, on the summit, right, on the upper part of the islands. And this area looks like this. In other words, it looks like treeless, no trees, more like shrubs, Meconia, grasses, ferns. Perhaps you can see some trees here, but these are introduced trees, introduced species, like avocado, for example. Yeah, but endemic or native species, they are not present here. This is a kind of an open niche, I'll say, an open habitat. Yeah, and there are no trees because this area is quite, it changes during the day. It can change from very warm, like 23, 24 degrees, up to down to maybe 8, 9 during the night. So somehow it's very difficult to get adapted to this zone. Also, humidity changing from dry to humid in one day. Now, regarding the geographical origins of the flora, we can say that most species are coming, I mean, the ancestors are coming from the western part of South America, from Venezuela to southern Chile. And here you have a table where it shows that more or less 56% of the endemic species, the origins of these species are coming from what we call the Andean zone. An Andean zone, it means, in other words, this western part of South America, as I mentioned before, from Venezuela to Chile. And regarding endemics, most of them are coming from the Neotropical region. And of course, Neotropical region is distributed generally in the tropics, okay? Now, regarding some endemic, native, and introduced species, this is very interesting because in Galapagos, we have more or less 33% of the vegetation is endemic, 180 species, and native 370. That means, in other words, 67% of the species are native. Now, I have two percentages to figure here, and this is much lower than that one. And that is because here I'm taking into account the introduced species as well. So out of over 1,400 species, endemics represent 12%, but if we do not take into consideration the introduced species, that goes up to 33%. Now, taking the opportunity of this table, we can see that regarding the vascular plants, that native plus endemics, the total number of species is 550, more or less. And that number is lower than 910, yeah, that are introduced species. In other words, there are more introduced species than endemic plus natives, yeah. So in other words, my message perhaps is go, I mean, come here to the Galapagos as soon as possible. Otherwise, in the future, you're going to see more or even more introduced species than endemics and natives. Okay, so anyway, so this is a very striking figure, you know, that there are more introduced than endemics and natives. Okay, but unfortunately, as we are going to see, institutions like the National Park Service and the Charles Darwin Research Station, among others, they are doing really good work, a job, and efforts in order to preserve this vegetation. And regarding the most diverse families in Galapagos or in general in Ecuador. Well, Ecuador, as you know, they are, perhaps you know, is divided in 4 geographical regions: Galapagos Coast, Andean, Amazon region. And these 3 regions are plenty of the Orchidaceae family representatives. And in Galapagos is the Asteraceae. Yeah, is the most diverse common family. That is because, as you know, Orchidaceae, the orchids, are much more difficult to spread from the mainland to the archipelago, because we have associations with fungi and on and on and on. Okay. So some of the endemic genera, I'm talking about genera. There are 3 families: the Compositae, Cactaceae, Cucurbitaceae, with endemic genera. Yeah, Darwinotamnus, Leucocarpus, Macraea, and Scalesia. See, this is very interesting. This genus is endemic, as I already said, and is represented by 16 species. Yeah, then we have the Cactaceae, Brachycerus, Jaminocerus, and Cucurbitaceae with Cysio-Caulis, which unfortunately is extinct. Some represent pictures of some of their representatives. The cacti, these 2 genera are endemic: Jasminocerus and Brachycerus, and that is Cysio-Caulis. Vascular plant, bryophyte, pteridophyte also are present there, but the endemism is much lower than with the vascular plants. And as I already said, the endemism is higher in the lower areas, in the what we call the arid zone. The humid zone, of course, there are some endemics, 26% and 4% in the littoral zone. Now, interesting fact is, for example, adaptive radiation is very important here. There are very interesting examples of adaptive radiation in the Galapagos flora. And remember, well, you know, the term adaptive radiation, right, is when one species or ancestor arrives from the mainland, arrives in the islands, and then different species are originated out of that ancestor. And finally, these new species are adapted to new or different open ecological niches. So in the end, you have species which are very different morphologically speaking, but not genetically. Genetically, they are quite similar. It's a similar example with, we have with in animals. With the Darwin finches, the beaks are different according to the seeds that they feed on. And in Galapagos, of course, we have different examples of adaptive radiation. Scalesia, the one that I already mentioned, plus other ones like in Chamaesyce, Califa, and Borreria. And Scalesia, as I said before, has been, you know, the opportunity to differentiate into more or less 16. This is under study, 16 different species. Yeah, and the ancestors in the mainland Ecuador, and here you have some representatives. And what's interesting, this is the archipelago, right? That some islands have their own species. Like, for example, let's take a quick example with this one. This is this island. This is San Cristobal, and San Cristobal, you can see, you can find 4 species. Yeah. To here, that one, and here. But these 3 species, Gordijoy, Divisa, and that one, are present in San Cristobal only. This one is Pedunculata. Pedunculata is present in other islands, like Santa Cruz, for example, Santiago, and other islands, okay? But what I'm trying to say is that here you have the different taxa, here the different islands, and some taxa are very specific in the presence that is present only in some of the islands. So very interesting. And also there is a quite interesting distribution of the species according to the altitude and geography of the islands. Some of them are present only on the lowlands, like for example, the example that we are taking here, San Cristobal, And in this situation, in this example, one of them is present on the highlands. And look at this. This is a really nice example. At least you can see the morphology, how it changes according to where the species are growing. In the lowlands, it's more like this with less surface, yeah, exposed to the sunlight. In other words, in order to avoid the evapotranspiration. Yeah. Now, just to finish now, then conservation issues. Very quickly, I already showed you that there are a lot of introduced species. Most of the introduced species are present in the inhabited islands, of course, and also there is a very, as you can see here, how the number of introduced species have been changing in the years according to also to the number of residents and tourists. And some of the most common introduced species, and they are invasive. Not all the introduced species are invasive, but some of them, more or less 3%, are invasive, like the guava, the red quinine, the mora or blackberry, lantana, yeah, elephant grass, Spanish cedar. These are very invasive species on the islands. And then this is very interesting, that there are some species that are congeneric. In other words, in one genus, you have a species which can be introduced and an endemic one. See, with Psidium, this is guava, the common guava, and this is the Galapagos guava, and lantana, this is the introduced, and this is the endemic. So in other words, what I'm trying to say with this is that if you want to apply something like biological control, you have to be very careful, because these two are quite related. And if you want to eliminate, for example, the introduced guava, yeah, in the end, you can become famous, because also if you don't take enough care, you can eliminate also the endemic one. See what I mean? So it's very important to do this by molecular work before applying any kind of biological control, which is forbidden by the way now in Galapagos. So some impacts very quickly. I'm going to finish 2 minutes. Impacts, see here, you can see the cinchona, very thick stands. Unfortunately, the park has been working on this and in eliminating with herbicides this vegetation. Here you have guava, the guava and the mora taking over the highlands, or the same thing with some of the introduced animals like the goats. Yeah, it's the competition for the natural herbivores, the tortoises. And this, well, you know, the goats, they are usually in groups, but not so the tortoises. And here are some of the changes that the goats can promote. See, in 20 years, in the same spot, the picture taken in the same spot, you can see how this can change. And here's just the rock, that's it. Yeah, but anyway, this is just to show you how introduced organisms in general can change the environment. And what I've been doing is fencing areas with endemic species in order to protect them from the goats, for example, in this situation. Of course, the idea is to have a seed bank. So in the future, what have been done, yeah, the goats, when they are going to be eliminated, the seeds can grow, you know, in new seedlings and a quick eradication of some goats with hunters, yeah, with help of dogs, also by a helicopter, by air. And so in summary, the introduction of a plant and animal species, of course, promotes alterations in native communities, in physical changes, alteration, native flora, erosion, extinction, local climatic changes. And of course, the idea is to protect the native vegetation, restoration of the vegetation, control eradication of animal plant, introduced species. And of course, in Galapagos, we do have a quarantine system. And sorry for, 9-10. Okay, so that's more or less what I have to share with you in a very quick time. Thank you.
Thank you very much, Hugo. I like to ask our colleague Andrea, she's been monitoring for questions and answers. And so please, Andrea.
Thank you. Thank you for your great presentation. We have a couple of questions. Sophia is asking, Galapagos Archipelago is an evolutionary living laboratory. Why the protection in order to avoid the invasive species is not stopped?
Yeah, it's a good question. The idea is this one. We do try to, no, well, not we, but institutions are there. They are very strong programs in order to eradicate and control invasive species. But the problem is that time-consuming and also, of course, very expensive. Yeah, and also there are some problems. Like sometimes, for example, in order to eradicate some plants, for example, some species, the problem is that sometimes people they do not want to get rid of some of these what we call invasive species, for example, guava, because the fruit, for example, are used in cattle. Yeah, they feed cattle with the fruits or shade. So it's quite difficult sometimes to work the local population because they do not accept this as a real invasive or something bad for the conservation. But of course, there are some programs, yeah, in order to control and even eradicate. Some species have been actually eradicated from at least specific spots, like what I showed you with the goats, for example. Yeah, but anyway, just to say, I have to say that there are programs with the objective of control and eradicate this species, but unfortunately it's time consuming and very expensive. Right now, with this, I'm going to finish the answer. Like with the blackberry, for example, it's in every single inhabited island, and it's terrible right now. So there are some studies carry on, you know, in trying to look for perhaps something like related to biological control in the future. Okay.
Thank you. Is there any case that the species dispersed back from the mainland to the mainland from Galapagos?
I understand. I mean, any case where from the mainland to the Galapagos, the species, yeah. I mean, I'm talking about the ancestors or the introduced ones. The ancestors, of course. I mean, there are some ancestors, yeah, that have been dispersed from the Ecuadorian, for example, or from the western part of the American continent to the Galapagos. And of course, you can look at the morphology, but not only the morphology, but strongly, you know, is studied with can show this relationship according to the molecular work. Yeah, and yeah, there are several examples where we can see that there are strong relationships between some of these endemics with at least one or two species back on the continent. Yeah, and is there a coevolution between endemic plants and native animals? I'm reading the questions here. Okay, is there a coevolution between endemic plants and native animals? Yeah, I say yes. Yeah, very interesting one. Just one coevolution is, for example, tortoises and one species, native species in the Galapagos. This native species produces a tiny apple, yeah, which is poisonous. Yeah, for every other living creature, with the exception of the tortoises, is poisonous. And it's very interesting because this species, but the seeds of this plant species had to pass through the digestive system in order to increase the germination rate. Yeah, so sort of, and of course, the tortoise uses, yeah, as a food resource, but also on the other side, this apple uses for, you know, increase the germination. And of course, in order to get dispersed, you know, over the islands, yeah. How do you determine that some species acquire adaptive radiation? Yeah, with molecular work. Yeah, you look at the ancestors, and also you can see how they are being adapted from the lowland to the highlands, for example. Yeah, the same genus with different species, but they have different leaf shape. They have different adaptations to humidity, to dry conditions. So what you have here in animal you have the Darwin finches. The beak is different according to the seeds where they feed on, right? In plants, you are going to have different adaptations, morphological adaptation, like different leaf shape or height, yeah, according to the humidity, according to the altitude, and on and on and on. Okay. Thank you very much for your wonderful talk. Could you give more information for the case in the Galapagos Island? How many invasive species are among the introduced plants? What are the potential effect of climate change? This is very interesting. Yeah, I'm going to start with the last one. What are the potential effects of climate change in the flora? We're studying this right now, and we're trying to set the baseline, what we have, right? And of course, we want to compare what we have right now with the future years. So scientists, what have been doing, establishing quadrats, yeah, and usually it's 20 by 20 meters quadrats. And so the idea is to see what's inside, of course, identify what is inside, and then see the changes throughout the time. At least for now, there are no real change, scientific data, but we hope to have that in the future. But of course, the climate has changed during these last years, the currents, the temperature has been changing. So in other words, indeed, we have some effects of the climate change. How many invasive species among the are there among the introduced plant? More or less 30 species out of 900. 30 are under this category of invasive species. In other words, not all the introduced species, plant species, are invasive. 30 of them. You say, well, just 30. Well, just 30, that's a lot, because some of them, like the blackberry, is very, very aggressive, I'll say. Could you give more information for TKC in the Galapagos Islands? There are some representatives, but they are more natives. Not endemic for the islands. They're more like native ones.
Hugo. Hugo.
Please,
Yes, I'm really
it's fine.
sorry.
It's fine. It's fine. No, it's fine. It's fine. Yeah,
yes.
I appreciate your interest in my talk, but we can communicate in the future. You have my email, and I'll be most glad to answer your questions. And thank you for your attention. Thank you. Okay.
Thank you very much, Hugo. If you please could type in the chat your email for people to have
sure. Okay,
yes.
sure. Okay. Bye.
Thank you very much.
Thank you.
Bye.
Thank you.
And we will continue with the following speaker. And I'm going to upload the biography, and then our colleague Dr. Xu Yu-gun will introduce her. So let me just open. Am I sharing it or not yet? No, not yet. 7.4. Share. So are you able to see the screen?
Yeah.
Okay.
Yeah. I'm very excited after listening to the talk about the Galapagos plant, very, very exciting. Now we return to China. Okay, Dr. Hui Liu is an associate professor in South China Botanical Garden. And she received her bachelor degree from Beijing Normal University, master degree from Institute of Botany, Chinese Academy of Sciences in Beijing, and then PhD degree from University of Sheffield, United Kingdom in 2012. She has joined the South China Botanical Garden since then after her getting her PhD, and then visit University of Arizona in 2018. Dr. Liu focused on plant evolutionary ecophysiology. Her note works include, first, illuminating phylogenetic niche conservatism in plant functional trait, a different plant clade, such as Magnoliaceae, Poaceae. And second, clarifying the spatial and temporal pattern of plant climatic niche, which explains the geographic distribution of plant species from a new perspective. Third, proposing the hypothesis of the coordination between hydraulic trait and water gradient, which expanded the frontier of plant ecophysiology. In recent 5 years, Dr. Liu has published 34 SCI-cited papers in scientific journals. A lot of them are highly cited. Her representative paper includes Science and Advance, Nature, Ecology and Evolution, New Phytologist, Journal of Experimental Botany, and others. The total citation of this paper reached 1,361. Oh, very high excited. She's a member of several ecological societies and a reviewer for many SCI journals. She has presented at many international and national conferences and received 3 awards for outstanding report. She's also keen on popular science and education. She was invited to be the editor of an essay journal, Plant Diversity, in this year. So his topic will concentrate now on distribution, evolution, and adaptation of plant functional trait of Magnoliaceae in China. Very good study. Please, Dr. Liu.
Thank you. Thank you. Hi, it's my great honor to be introduced by Professor Ge, and thanks for Alina for all her efforts for organizing the meeting. And now I'm sharing my screen. Can you see and hear my PowerPoint?
Yes.
Okay, let me try.
Buenos amigos. My name is Hui Liu. I'm from South China Botanical Garden. Today is my great honor to be invited to join Chinese-Neotropical Botanical Diversity Conference. And thanks to Professor Xuejun Ge to give me the opportunity to share some of my work on Magnoliaceae. Today, my talk will focus on distribution, evolution, and functional adaptation of Magnoliaceae in China. Here is my outline. First of all, background. When we talk about the species evolution and distribution, we always use the word phylogeny to describe species relationships and geography to describe species distribution patterns. Ever since 1859, Charles Darwin published his famous book, The Origin of Species, that people start to realize all the species on Earth actually have the same common ancestor. Also, people found the closely related species tend to share similar niches and traits. This also happens in plant kingdom. There are 37,000 plant species on Earth and belong to algae, ferns, gymnosperms, and angiosperms, etc. These show astonishing diverse leaves and flowers. However, people can still distinguish them and classify them into nearly 500 different families because they show both similar or distinctive traits of the same organs. For example, the flower traits. Taxonomists always use them to distinguish species. This will bring a question that how conservatism of these traits. This is called a phylogenetic niche conservatism, PNC. Here, the figure on the left shows a phylogenetic tree and the trait values, the bar. Currently, we use Pageau's lambda and Blomberg's K to describe the degree of PNC. Lambda has a value from 0 to 1. When lambda equals 0, that means all these traits are randomly distributed across the tree. When lambda equals 1, this trait values follow the pattern called a Brownian motion model. That means this trait follows the evolutional pattern. And Blomberg's K has a similar meaning, but the value ranges from 0 to 1 and above 1. Similar thing that when K equals 0 means trait is randomly distributed. When K equals 1, it follows the BM model. PNC has been widely proved across different traits and from both plants and animals, from both from the species level to the community or the regional and global scales. However, which traits are more conservative is still unclear. Phylogenetic biogeography is used to link evolution to species distribution. Because these species, they evolve in different regions, they will have different traits or the different genotypes so that they show both divergent and convergent evolution patterns. Plants will have used functional traits to adapt to different environments. When we talk about functional traits, we briefly use morphological, physiological, and phenological traits. For example, I'm doing hydraulic studies. So plants absorb water from soil, transport them through stems, and transpire it through the stomata. And they fix the carbon dioxide to the leaves through photosynthesis. During all the stomatal or the stem or branch trees, we also focus on trait-trait coordination, such as the water transport process and the photosynthetic process. About magnolia, first of all, it's a very basal angiosperm family that originated about 100 million years ago. It has lots of primitive morphological traits and pollination styles. For example, here, they don't have the difference between petals and sepals. They only call the one organ called perianthely. Perianthely, they have non-perianthely that have arranged in 3 layers to form their flower, and they have many spirally arranged stamens here. Some of Magnolia species have very large leaves, which is ancient leaf style, and they are pollinated by beetles rather than bees because about 100 million years ago, there are still no bees have originated. For Magnolia, they have another special thing is that they have seeds outside their fruits rather than inside. Magnolia species has very beautiful flowers and loved by many people around the world. However, nearly half of the species in this family are in the IUCN red list here. And currently, they distribute mainly in Southeast Asia and North or South America here. magnolia species are very valuable, not only because they play important roles in the subtropical or tropical forests. Some of them are dominant species. They also have important values for herbal medicine or the perfume production. They're also important horticulture species. You can buy magnolia seedlings from website and grow them in the garden to have beautiful flowers in the spring. People in China love magnolia species very much. It's kind of a culture symbol in Chinese history. For example, people make poems about magnolia species, and they put magnolia flowers on the skills and everything they use in their daily life. Now, climate change threatens many species and also plant species in the world, especially for this Southeast Asia and South America. For these hotspots, they are just where the magnolia species distribute. Many of them are threatened by field exploration or pollution. Therefore, we built the largest magnolia garden in South China Botanical Garden here. We conserved nearly 200 species, magnolia species here. And here also the location of Magnolia Society International. We do a lot of progress on both axial and in-shield conservation of this beautiful family. Now let's move to the phylogeny of magnolia. Currently, there are two taxonomic systems here. One is to describe to have two genera. One is called a Liriodendra here. The other just called magnolia. I used the taxonomy system proposed by Xia Nianhe, professor from South China Botanical Garden. He divided the family into 17 genera listed here, according to their different morphology traits. However, these two taxonomy systems are actually share the same phylogeny traits. It's just the name, the general name, be changed into section names. Here, the numbers are the species number exists in China. You can see there are a lot of species numbers 0 because they originated from South America. This is also another reason that I give this talk to share more information with you, with people from universities and institute in neotropics. We can share more information about this species conservation and scientific collaborations. Yeah, about the distribution of magnolia, it's a typical historical continental disjunction, as I showed before. The distribution center has over 18% species in Eastern to Southeast Asia. But the origin center, where the first fossils are digged out, is in temperate area. So the real original area is still unclear. Given all these knowledges, we propose our questions that we have the phylogenetic tree and their distribution patterns. We have environmental variables. Then we go to these places to measure their functional traits to see how they adapted to these different environments. So our main question include first, we see how this phylogeny and special drivers in trait variation. And the second study, we try to look at the phylogeny signal, the produce lambda in functional traits, and how they correlated involve between trait pairs. And the third study, we go further to see how functional traits adapt to different regions and seasons. So You can see the logic between these studies. So we want to know how they distribute the geographic divergence. And then we try to see how this tree distribute across the phylogenetic tree, how those trees are conserved. And we also go to different regions to measure their functional adaptations. So here is study one, focus on the distribution. We try to distinguish evolution and spatial drivers in tree variants. We first collected all the species distribution places and maps them here. Here are the 4 largest lineages in Magnoliaceae. It shows a clear temperate and tropical divergent distribution. Here, the Eulalia is centered in temperate area, but Magnoliaceae, Michela, Lawrenceae, and Magletia, they all distribute in South or Southwest China. Based on their distribution, we can build a model that to divided the trait variance into 3 parts. The first part, we don't need to go to details about this model. You can have the idea that lambda is just a lambda I just say. It means the effect from phylogeny, and phi focus on space, this distance, and then a residue that independent of both. So this is our first model. It's called a philo plus spatial model. And then we build another model to fix lambda equals 0 and phi equal 0.99. It's called the spatial only model. And then we change it to no spatial factors, calculate the real lambda value phylogeny only model. And finally, we use another non-model that follow the pure BM model. So we compare the 4 models for different trees to see the relative contributions of evolution in space. Our results show that plant trees here, they have the best model from the pure evolution model. And for the environmental variables, both phylogeny and space, they matter. So let's give a summary that the variance of most plant structure traits were explained more by phylogeny, whereas variance of environmental related traits were associated more with space. Then we move to see about the evolution of all those traits. We try to measure their phylogenetic signal and how they evolved. We do our experiments in Magnolia Garden here. We choose the 34 species from the garden and measure the 20 functional traits that as I introduced, including hydraulic and photosynthesis. So we find a strong phylogenetic signal in most hydraulic and morphological traits, like the very high near 1 for the wood density. And we found a weak phylogenetic signal in photosynthetic, but not for these nutrient-related ones. And for the correlated evolution, we use a model called the phylogenetic generalized linear model. This line will have phylogeny considered, so that this line is always exists. Means the pair for the tree, for the 2 trees, will exist independent of phylogeny. Here, the branch is hydraulic conductance, how fast the water moves through the branch, and the wood density. That means if we have a very dense wood, you will have a very narrow vessels so that you can transport water at a low efficient. This relationship will always exist, give phylogeny considered or not. And this similar thing happens for leaf trees. Later we do a phylogenetic principal component analysis, which try to divide those species into groups to see which trait drive this pattern. We found that the Larianceae is separated from the other 3 genera because they have a very dense wood and dense leaves. Here we can show the phylogenetic signal. Here you can see this is the 4 largest genera, Michella and Magnolia. They have very high tall trees, and they have very dense wood, as I just showed you. For Eulalia, I put it in blue because it's centered in from the North China. They have a high nitrogen and photosynthesis rate and high hydraulic conductivity. For Magnolia, they also have higher hydraulic transport ability. So let's give a summary. So we found the phylogeny signal in hydraulic and nutrient-related traits. And we found the correlated evolution of several key functional traits. For tropical evergreen lineages, that they use storage at a very drought-tolerant trait, but they can have a low photosynthetic rate. However, for temperate deciduous species, they grow fast. They have high hydraulic transportation, but at the cost of their drought tolerance. The third study is about the adaptation in different regions and seasons. We do this study because we found that these 2 genera have a very contrasting distribution. One is in South, the other is in North China. We choose the Botanical Garden from South China Botanical Garden and Wuhan Botanical Garden, located in the distribution center. And you can see they have a very different climate, but they both have the wet season and dry season. During the wet season, the temperature in Wuhan is even higher. But for in Guangzhou, here in South China, the temperature in winter is higher, but the precipitation will be similar in drier season. But in South China, we have a very high precipitation. We select the 4 common species that grow in both gardens. 2 are deciduous species, and the other 2 are evergreen species. We found plants in North Center, they face the more drought stress in the wet season. Here, the white bars, the more negative this per dawn water potential means the species suffer more drought stress. And the same for the midday water potential. The insert figure shows the data in dry season because deciduous species, they don't have leaves, so we can only measure these evergreen ones. They also show a not so clear pattern here. We found plants in North, they had a lower stomato-related traits, including photosynthetic rate and stomatoconductance, and a lower stomatopore area index. This means how much stomato area that per leaf area. The higher the value means the higher leaves can transfer water through stomato. No, but for the dry season, the only stomatoconductor differs, but not for the other 2. Another interesting result shows that deciduous plants in North, they had a lower hydraulic traits, but not for evergreen ones. For example, here, is the branch hydraulic conductivity and leaf hydraulic conductivity. The same species in salts, they will show higher values for this, but not for the other. We don't find any differences in leaf and branch structures. For example, the specific leaf area and all branch wood density, they are always the same, even from dry season or wet season or from South China. A brief summary. So magnolia species, they adapt to regional climate mainly through some of the trees. Other trees show limited plastic responses. And second, deciduous species are more sensitive to regional difference than evergreen species. Dry season seems not affect the growth of actually conserved magnolia species. We are still looking for the reasons for this pattern. And finally, the acknowledgement. First of all, I'd like to thank all the people that helped me during all this work, especially for Professor Qing Ye in South China Botanical Garden and Dr. Chu Yuanxu, who did a lot of work in measuring all these functional trees. And Marginal Langer from University of Lancaster, and also Robert Frankleton from University of Sheffield. They do all these phylogenetic models with me. And Luis Santiago from University of California, Riverside. He gave a lot of advice on the PNC measurements. And Wang Yuhao from Institute of Applied Ecology, Chinese Academy of Sciences. He's expert on marine hydraulic trees and many others. I also want to thank the staff in Magnolia Garden in South China Botanical Garden. They work years to take care of all the magnolia trees grown here, and they exchange species from different gardens all over the world. They are Xin Lanchen and Ke Yang. I also like to thank all the members from our group. We are a good team to do all this scientific work. Finally, I'd like to thank funding National Science Foundation of China and the Chinese Academy of Sciences and our garden and the Youth Innovation Promotion Association of CAS, and also from Guangdong and Guangzhou Science and Technology Foundation and China Scholarship Council. And welcome to South China Botanical Garden. There is also a great news I'd like to share, that on 11th July 2022, South China Botanical Garden is upgraded as South China National Botanical Garden. Currently, there are 2 national botanical gardens in China. One is in Beijing, Institute of Botany, where I graduated from, and now the South China National Botanical Garden, where I work in. They will be the 2 leading institutes in the species conservation, ecology restoration, and the carbon neutralization, etc. Finally, I'd like to share a video that will celebrate the unveil ceremony. Although it's in Chinese, I will briefly translate some information for you to look around our garden.
高知图南，巍巍理想，孕育着万千风华的岭南大地，生机盎然。中国是植物多样性最丰富的国家之一，人类地球家园37万余种生物中。
China has over near 14,000 species plant species here.
滋养了人与自然相互生活的东方文明。 10月12日。
In 2020, our President Xi
北
Jinping
广州等国家植物
started
园
the National Botanical Garden exhibition
2022年5
on
月40日，
both Beijing and Guangzhou.
在广东省广州市设立华南国家植物园。广州地处全球生物多样性热点地
Now
区，
it's the landscape in
是被玫
Guangzhou.
瑰介绍的美丽荒城。
Guangzhou is called a flower city.
坐落于自的中国科学院华南植物园，由著名植物学家陈焕宇院士创建于1929年。新中国成立以来，在多位党和国
This
家
is
领导
the
人
history
一起
of
关怀
our
之下，
botanical
首
garden.
代科学家和城市
There
设计
are,
者
I think, over 300
华南植物
scientists
园
here.
成为中国最佳植物园之一。综合排名位居世界前列。
And in 2019, we are ranked for the first 10 botanical garden around the world, I think.
棕园、江
We
园、
have 38
38
gardens for the different lineages.
青地保育华南地区80%以上。
And we conserved 17,000 plant species from South China.
在植物分类学、恢复生态学、植物资源保护利用等领域，已建成具有国际水平的重要高地。纵观人类文明发展，生态性则文明性。试图贯彻习近平
These
生
are
产文明思
our
想。
garden.
华南国家植物园建设将坚持以华南地区植物栖地保护、高水平研究、可持续利用
We will keep focusing on actual species conservation and ecological restoration and
建设种植资源库、植物栖
the
地保护
species
区利用平台、
seed conservation and
资源植物
public education,
分类员，
etc.
探索国家植物园管理运
This
行
is our herbarium.
紧密开展全球合作，筑起中国生物安全和生态安全屏障。
This is our field station.
讲好中国植物故事，彰显中华文化和生物多样性魅力。广州将以华南国家植物园建设为契机，大力探索成员共融，传承发展岭南园林文化，构建布局均衡、全民友好的公园体系。 在广州，不同人物拥护山林之谊，生于闹市，可讲林泉之意，让绿色低碳融入生活，公园家园毗邻城景，文明意识扎根民心。为粤港人文湾区建设，广东、广州城市高质量发展注入强劲动力。 在习近平生态文明
We
思
will
想
combine the garden into the city construction to build a harmony in society.
人与自然和谐共生的绿色大球，为构建地球生命共同体贡献中国智慧、中国力量。
All right, that's all. And thank you. Anyone who want to collaborate with me, please send me emails. Bye bye.
Okay, since I'm sharing my screen, I can't see the question. Let me try to open.
Julio, maybe
Do you hear me? Okay.
Yeah.
Alina,
can
please.
we? Yes, very neat. Thank you. Maybe before we start with the questions, you could continue with your presentation of the of the
Okay,
journal?
sure.
Yeah.
It's just show people that. Oh, sorry. Yeah, can you see this? Yeah, currently, the Journal of Plant Diversity is from Kunming Institute of Botany. The journal has the impact factor 3.3, and it's an open access journal, but it's free of charge. And let me check. Yeah, it focuses many multiple discipline researches. For example, phylogenetic, taxonomy, evolution studies, reproductive biology, biogeography, population biology, conservation biology, molecular evolution, paleobotany, and physiology, etc. So welcome everyone who study of researches related with biodiversity. We think the journal is very quickly have the review process within 2 months, I think. And we are promoting these journals, and thank you. You can check any information from its main page. I think that's all. Now let me check the question. Is this the first one? Do factors below the soil influence evolution among species, like nutrient difference, nutrient deficiency? Thanks for Sadium. I think nutrient factor is short-term influence, but it depends on the skill. For example, if you compare Eastern China and Western China, like Professor Li Min just showed, the large scale soil nutrient difference is East China has a better nutrient, but Western China has some poor nutrient soil that will definitely influence the evolution process. But for the short-term scale, for example, 100 years, the soil difference will not influence the evolution process. That's my viewpoint. And the second question, is it possible that Magnolia's ancestor is older than 100 million years? Yes, because 100 million years is just approximate estimation, because some molecular ecology studies show that Magnolia's ancestor may be 100 or 140 million years ago, but this is still discussing and not a fixed time. It's very illegitimate and hard to understand by containing only this purposes. Yes, but thanks for Sophia's question. But I, because I'm major actually in ecophysiology, I didn't do this evolution and speciation study, so I can't answer you from the evolution perspective. But I can see this is people are still studying this difference and the arranging of the Magnolia, because it's really a very basal angiosperm garden. Oh, thank you. The beautiful garden. Yes. Thanks from question from Xiao Huajin. Do the phylogenetic conservation have efforts on diversification in Magnolia? This is a very good question. I think this is a two question. The phylogenetic conservatism focuses on the trait or niche differences across the species. It's basically based on the hypothesis that species are already there. So we measure this difference among these species. But diversification means you have some new species involved. And from a long-term scale, we can see that because currently some family they have fewer species, but some others family they can diversify at a very high rate and have many species within the family. But for magnolia species, they actually is a small family because they only have 300 species. I don't think phylogenetic conservatism they exist in this family, but they are not have the direct effect on the diversification process. I think these are two things. Do you think you understand me? And yeah. Are there any more questions?
Yeah, there are several questions. I don't know if Andrea is here or if there are more comments.
You already read the questions and answered them.
Yes.
Yeah, we don't have any more questions on the chat or in any other.
Yeah. Thanks.
Thank you.
Yeah.
Thank you.
If you have chance, just visit us. It's really beautiful here.
Okay, so we would like to thank
Stop sharing, right?
Yes, if you, yeah, good, wonderful. So we are, it's very nice. We've been on time, which is wonderful. So the next step in order to finish this 2-day symposium is we are going to have a closing event, a very short one. So for that, according to our program, I'm going to share. Yeah, we'll have first a presentation, a short video from Yang Yu, a collaborator from South China National Botanical Garden. And then we would have a few closing remarks from Andrea Andrade, who is the director of the tourism department at Universidad Regional Amazónica. Then we will have the words from Dr. Roldan Torres, who is the vice chancellor of research, I'm sorry, of academics at Universidad Regional Amazónica ICAM. And then Xu Jun and myself would just give a few words of thank you and a few words for talking about possibilities of collaboration. So I'm going to share, I will try to share the video from Magister Yang Yu. I hope the audio will work. If it doesn't work from my computer, then Andrea is going to help us sharing the video. So let me see. Closure. Are you able to hear? Were you able to hear the recording?
You're not sharing a link.
I can only hear you. I can only hear you, but not the video.
So you can hear. I just wanted to make sure that you hear. So now I will share.
Yes, we can.
Can everybody see the, yeah, you can see the, okay, so let's go. Sorry. Yeah.
No audio, Aline.
No audio. Okay. Then I'm sorry, I will ask Andrea, please, to share. I don't know why I'm not having audio. Please.
Hi everyone, this is Yu Yan from South China Botanical Garden.
The audio is not working.
Chinese Academy of Sciences. I'm in charge of international affairs. It's my great pleasure on behalf of SCBG to take the chance to speak on our symposium. First, let me offer my warmest congratulations and my sincere appreciations to all organizing committee members. I know how much hard working and how much patience needed to do for a symposium. Thank you, Alina, Professor Ge, Andrew, and Tamia. Second, I will thank all of our speakers. During the 2 days, we have a pleasant journey from aquatic plants, coastal plants, alpine flora, even from the Pyramids to the Galapagos. Thank you so much for bringing us your impressive research progress and sharing with us. Finally, I would thank all of the participants from Asia, Africa, Europe, and the Americas. Your journey inspires us a lot. I hope this symposium will be a good start for our future cooperation. We can do the co-organized project, symposium, workshop, conference. We can have joint supervision of graduate students, partnership groups, joint laboratories, and joint research centers. We even can exchange our scholars, specimens. Many things we can do. So, we are waiting for you in SCBG.
Okay. Thank you, Andrea. So now it's my pleasure again to introduce to Andrea Andrade, who is being a member of the organizing committee, who is also the director of the tourism department, and she is from Universidad Técnica de Cotopaxi, one of the other organizing institutions. So please, Andrea, welcome.
Thank you, Alina. Thank you, everyone. This has been 2 days of a lot of sharing and knowledge experience from different parts of the world, especially Ecuador and China. America, we can see that diversity is very important part of everyday life, I could say. Sometimes we do not see what is beyond our own eyes, and these kind of events help us to show a little bit of what we have, what we're looking at. I hope these talks that we have, our speakers that put lots of effort on giving us some of the most important information they've researched through the years, has helped us to encourage on keeping doing these investigations, working on botanical studies on Ecuador, the Amazon, the Andean region, and everywhere we are. This helps us, as Alina said, like in some places, to see the potential uses that all diversity has. So I would like to thank, because also my students have been part of this, even though they do not completely understand English, they've been happy to listen to your talks. And I know even a bit they might have understood during these 2 days, have been a lot of information for them, and it's been a great experience for all of us. Thank you, everyone, for giving this space to us and for sharing everything that you've done. Thank you for the people that have helped us through all these months to put together this event, and sorry for any inconvenience that we have had through the technology. Thank you again, and that will be, thank you.
Thank you very much, Andrea, who was talking on behalf of the authorities at Universidad Técnica de Cotopaxi. So now I would like to welcome Dr. Roldan Torres, who is the academic vice chancellor at Universidad Regional Amazónica ICAM, which is my institution, and it's also one of the organizing institutions of the symposium. So Roldan, thank you and welcome.
Thank you very much, Alina. Can you hear me? It's okay? Yeah. Thank you. Thank you very much. It's a pleasure for me to be here in this closer event, this important event for us, for our institution, mainly for ECAM. And I would like to start giving my grateful and also my really very thank you to Alina for the coordination of this symposia that is very important for our institution, for Ecuador, and also for the relationship between our institutions. These topics that you have discussed during these days are one of the most important in the basis to reinforce the research and also for teaching and for academy in the whole world topics. For us, we are located in Amazon region, and therefore to reinforce the importance of this kind of events and symposia, because it's one way that we have to open the border, and the collaboration among universities is really important and most in this kind of topics. All the presentations and the talks that you presented during these days are really important also, and I want to give all my thanks also to all the speakers and all the organizing committee that has done possible this kind of symposia. Finally, I want to open the doors of ECAM for the collaboration and also for to do this kind of symposia more often, because we need it. We need to reinforce our collaboration and this relationship between researchers are really important to increase and to improve our skills in this kind of topics, and also to involve in this topics and in this symposia our students that are the main reason of our university. Finally, to all the organizing committee, to all the people that were involved in this symposia, I would like to thank also for the name of our rector and the people of our institution that were related to the symposia. Thank you very much. I hope that this kind of events can do presentially, and ECAM is open to receive with open arms to receive all of you in our institution, and to have the real life of the Amazonian, and to see the richness of the botanical issues in our territory. Thank you very much. Thanks, Alina, once again.
Thank you. Thank you very much, Roldan. So now we are going to participate. I like to introduce again, we've been like working also as Dr. Xu Jun, who is an investigator at the South China National Botanical Garden, and who is being my sponsor in the Presidential Award Fellowship from South China Botanical Garden and Chinese Academy of Sciences. So Dr. Xu Jun, please, if we could have a few words from you, and after his words, I would just like to say a few more words with thanks. Please, Xu Jun.
Hello, everybody. I'm very happy that this meeting was held successfully as expected. Thanks all of the people, Andrea and many others who help us. So I think the purpose of this meeting is to bridge the gap between China and the people in South America, because it's so far and the exchange in the past years is very few compared to our extensive exchange between China and the USA. So we hope through this meeting to enhance the communication between Chinese botanists and the South American botanists. Through this meeting, we know there is a PI-FI program. The PI-FI program its purpose is to push the cooperation between Chinese scientists and the foreign scientists. It includes from the PhD student, postdoc student, associate professor to full professors. So it covers all the range of researchers. For the student, from master student to PhD student, we have a university. It's called UCAS, University of Chinese Academy of Sciences. And for good student, the UCAS will provide scholarship to cover their study and living cost in China. It's not difficult to apply. So we hope and we encourage the good student to apply for the scholarship to UCAS. And also, we hope through this meeting, the audience could find the researchers in China that share the common interest. So they could to have exchange after the meetings. And we hope through the common interested scientific questions that they can produce or push some common research, how to say that, to fund research cooperation opportunities so that the cooperation could last longer. For example, through this meeting, I found I'm very interested in the Galapagos adaptive radiation. But unfortunately, I just found that the genome paper was published this year in Nature Communication. So that means that we know very little about each other. Although in the past 10 years, I have visited many times to South American countries. But in fact, for me, I know very few about Amazon forest, very few about high Andes, and I have never visited Galapagos Island. So there's a lot of opportunities for us to cooperate in future. We hope through these meetings to bridge the gap, to create opportunities for scientists from China and South American countries so that they can find opportunities to cooperate in future. So that's the purpose of this meeting. Thanks all of you to attend this meeting. Thank you.
Thank you very much, Jun. It's been a very nice event. And I would like to thank, so if you please spare me, it's the first, we would like to thank for our very important guest speakers for their sharing, for their generosity in sharing their knowledge. At least for me, I feel like it has been an intellectual treat, and I do thank you. So to Doctors Wang Qifeng, Javier Cornejo, Xin Yeo, Katia Romoleru. Unfortunately, Dr. Neil was unable because of medical issues. Lu Limin, Hugo Valdebenito, and Liu Hu. Thank you very much. We would also like to thank to the authorities and collaborators from the organizing institutions, South China National Botanical Garden, the PI-FI CAS President's International Fellowship Initiative, Universidad Regional Amazónica Iquiam, Universidad Técnica de Cotopaxi, and Asociación Botánica Ecuatoriana. As also, we'd like to thank the participating institutions, South China Botanical Garden, Universidad Estatal Amazónica, Institute of Botany at CAS, Universidad de Guayaquil, Wuhan Botanical Gardens, CAS, Xinhuangbanna Tropical Botanical Garden, CAS, Universidad San Francisco de Quito, and Pontificia Universidad Católica del Ecuador. And how do you say lastly, but not leastly? Well, also we would like to ask very much the organizing and collaborating committee members, in particular, Dr. Xu Jun, Yang Yu, Andrea Andrade, Galia Rives, Lisette Carrion, Samia Salazar, Daniel Martinez, Nadia Zurita, the NOC CDA team, and the communications and technologies teams from these organizing institutions. And of course, the attendees, you were very important. So thank you for your patience with these little hiccups we had. And thank you very much for everybody, and we hope to see you someday personally somewhere.
Yeah.
Thank you, Alina.
So think if anybody wants to say a few words, or we could just finish the meeting.
Thank you very much.
Okay. So thank you, and see you around.
Yeah, see you in the future. Yeah.